Welcome to Sartor TV, an idea-sharing platform that was founded by Russell Sartor, who's an author, investor, and the CEO of Netcom Learning. Hi, I'm Vaishali Jain, and today we're excited to bring to you a special episode we created with the New York Hall of Science. It will offer a deep dive into the revolution of bioengineering and genetics being made possible with CRISPR gene editing technology, led by a panel of experts who will guide us through the policy and ethical implications of CRISPR on global health. Enjoy.
Good evening, everybody. I'm Margaret Honey, the president and CEO of the New York Hall of Science, and it's my pleasure to welcome all of you to our 5th annual Spring for STEM discussion. Tonight's program, we're really excited about it, features an extraordinary panel of experts who will explore the revolutionary gene editing technology known as CRISPR. But before I get started, I'd like to say a few words about NYSCI and our President's Council. You are scientists, business leaders, entrepreneurs, philanthropists, and others who have an interest in science, in education, and in the cultural and intellectual life of New York and the nation. And you provide crucial support to the museum, for which we're very, very grateful, driving our work as a laboratory for learning, a training ground for young scientists, and an anchor for members of our community. We thank you all who are already members and welcome you who are not yet to learn more and to join us. So I don't need to tell this group what science is and why it's important, but I do want to take a minute to talk about our distinctive approach to science learning and how we help our audiences to experience the real thing, the kind of deep knowledge plus serendipity that Siddhartha Mukherjee calls informed improvisation. We call our approach design, make, play, and we see every day how it enables young people to develop critical thinking, knowledge integration, and innovation skills, and that all-important sort of sense of agency, that sense of I can do this, in ways that are measurable and scalable. Especially today when we are no longer knowledge constrained and when textbooks make a whole lot less sense as teaching tools. Science is most fundamentally about pursuing questions that matter. So we do invite you all to learn more about how we do this. And when you come to NYSCI, we'll tell you about how our museum and our community anchor everything we do. Our work in family and public engagement in STEM, our youth development programming and career ladder, our work with the formal side of STEM education, and of course, our research. We'll tell you about the partnerships that extend our impact locally and around the world, and we'll show you how we do this in the most ethnically diverse county in the country. We are passionate about developing and nurturing the science innovators of the future. So thank you all for being here tonight and joining us on this journey. Now, on tonight's program, we are thrilled to have with us experts and leaders in biology and genetics who will help us understand the extraordinary gene editing technology, CRISPR. Before inviting our panelists to come up to the stage, we're going to play a brief video, so you're going to have to turn that way to see it, that helps to explain kind of the science underlying this technology.
Every cell in our body contains a copy of our genome, over 20,000 genes, 3 billion letters of DNA. DNA consists of 2 strands twisted into a double helix and held together by a simple pairing rule. A pairs with T and G pairs with C. Our genes shape who we are as individuals and as a species. Genes also have profound effects on health, and thanks to advances in DNA sequencing, researchers have identified thousands of genes that affect our risk of disease. To understand how genes work, researchers need ways to control them. Changing genes in living cells is not easy, but recently a new method has been developed that promises to dramatically improve our ability to edit the DNA of any species, including humans. The CRISPR method is based on a natural system used by bacteria to protect themselves from infection by viruses. When the bacterium detects the presence of virus DNA, it produces two types of short RNA, one of which contains a sequence that matches that of the invading virus. These two RNAs form a complex with a protein called Cas9. Cas9 is a nuclease, a type of enzyme that can cut DNA. When the matching sequence, known as a guide RNA, finds its target within the viral genome, the Cas9 cuts the target DNA, disabling the virus. Over the past few years, researchers studying this system realized that it could be engineered to cut not just viral DNA, but any DNA sequence at a precisely chosen location by changing the guide RNA to match the target. And this can be done not just in a test tube, but also within the nucleus of a living cell. Once inside the nucleus, the resulting complex will lock onto a short sequence known as the PAM. The Cas9 will unzip the DNA and match it to its target RNA. If the match is complete, the Cas9 will use two tiny molecular scissors to cut the DNA. When this happens, the cell tries to repair the cut. But the repair process is error-prone, leading to mutations that can disable the gene, allowing researchers to understand its function. These mutations are random, but sometimes researchers need to be more precise, for example, by replacing a mutant gene with a healthy copy. This can be done by adding another piece of DNA that carries the desired sequence. Once the CRISPR system has made a cut, this DNA template can pair up with the cut ends, recombining and replacing the original sequence with the new version. All this can be done in cultured cells, including stem cells that can give rise to many different cell types. It can also be done in a fertilized egg, allowing the creation of transgenic animals with targeted mutations. And unlike previous methods, CRISPR can be used to target many genes at once, a big advantage for studying complex human diseases that are caused not by a single mutation, but by many genes acting together. These methods are being improved rapidly and will have many applications in basic research, in drug development, in agriculture, and perhaps eventually for treating human patients with genetic disease.
So I'd like to ask our panelists to come up. So we're joined tonight by Sharon Begley, a senior writer at Stat News. And Sharon is really one of probably the leading journalist in the country that has covered CRISPR and other leading edge science topics. Sharon has graciously agreed to moderate the panel for us tonight, and she has the honor of introducing everyone. So Sharon, I'm going to turn things over to you. Thank you.
And thank you for having me. And let me add my welcome. It's great to see you all here this evening. So my job is fairly simple. We're not going to break any new ground in panel presentations here this evening. Our three speakers will each talk for several minutes about their work. And when we were organizing this, we asked them all to please give you all their CRISPR origin story, how they got involved in this field. And then I'll ask a few questions, probably myself, but we want to leave a lot of time for questions and answers. And there will be a pop quiz on the video, so I hope you've all paid lots of attention. I know you have their bios in the programs, but in case you don't want to take out your reading glasses, let me give you a short version. And we're going to start with Rachel. Rachel Horowitz is a co-founder of Caribou Biosciences, where she's been the president and CEO since its inception in 2011. Rachel is also a co-founder of Intellia Therapeutics. In 2014, she was named by Forbes magazine to the 30 Under 30 list in science and healthcare. And in 2016, Fortune named her to its 40 Under 40 list. Your own personal origin story as it relates to CRISPR, and then of course what Caribou is up to.
Absolutely. Well, good evening, everyone. It's such a pleasure for me to be here with you this evening. It may be hard to believe, but I am a CRISPR old-timer. I have been working on CRISPR since 2007. And as the video outlined, the earliest work in the CRISPR field really came from a very esoteric study of how bacteria and archaea fight off viruses and other selfish pieces of DNA. And so I was involved in that field for several years, and I worked at the University of California, Berkeley, in the laboratory of a woman named Jennifer Doudna. I was the first student in her group to be studying these proteins, trying to understand how they functioned and how they ultimately worked in concert to fight off viruses and allow many different kinds of bacteria to survive. Some of my colleagues in her lab happened to be working on the Cas9 protein that you heard about in the video this evening, which of course holds tremendous potential for genome editing and genome modification. And so I was lucky enough to be part of a group of scientists at UC Berkeley who wanted to found a company, create a space to try to translate a lot of this research into the commercial world. Also lucky enough that all of my co-founders are academics through and didn't want to leave the laboratory. And they gave me the opportunity to try to build and grow the team. And so at Caribou, we've been working on CRISPR gene editing for almost 7 years now. And today, we're primarily focused on developing new medicines using this technology. And we work in 2 different kinds of cells to do that. In part of our work, we modify immune cells, such as T cells or other part of the immune system, using gene editing to tweak these cells to actually teach them how to find and fight cancer. There are versions of these kinds of therapies already approved. In fact, the first ever cell therapies approved in the United States are an earlier version of this type of medicine, and they're called CAR-T therapies. But the versions that exist today, while miraculous for some of the patients who benefited from them, are very challenging to make. It actually requires clinicians to take a cancer patient's own immune cells, their own T cells, out of the body, into the laboratory, manufacture one product for that one patient, and then ultimately redeliver it back to that patient. It's very challenging. These are very ill people. These cells are not in great shape to begin with. And unfortunately, some of the patients are not even able to live long enough for the manufacturing process to be completed before they're able to receive their therapy. And so at Caribou, along with many other groups, we're working on ways to use gene editing to create the next version of these cellular therapies that can be so-called off-the-shelf, meaning they don't have to be made from a patient's own cells, but with the use of gene editing, we can delete the parts of the cell that would otherwise cause graft versus host disease or some of the other stumbling blocks that currently prevent the field from making these medicines more broadly available. The other part of our therapeutic focus is what we call bugs as drugs. And that is by focusing on the microbiome, which is the collection of microorganisms that live in and on us. And we're particularly interested in some of the bacteria that live in us and in our guts, and the role that they play in how our immune systems fight cancer. And so you can tell that there's a theme in our work of really focusing specifically on cancer therapies. And we're also using gene editing to manipulate microbes that live inside of us in order to better fight cancer. I will highlight, we do get to work outside of the human healthcare space as well. Gene editing has the potential to impact a number of very broad different industries, such as food, agriculture, basic research, and industrial biotechnology. And through licensing and business development, our organization has partnered with a number of companies who use CRISPR gene editing in other spaces, such as plant breeding and even livestock breeding. So one of the projects that we're working on right now with a British company called Genus PLC is actually breeding pigs that are resistant to a particular virus. There's one virus, PRRS is the acronym for the name of the virus, that's the worst problem that American and European pig farmers deal with. And it's an unfortunate situation where if one animal gets sick, basically every animal on the farm will get sick. They're too sickly to then go into the food stream. They all have to be slaughtered. And part of what makes them sick prevents them from reproducing. And so all of the animals are killed, they're not going into the food stream, and a farmer has to completely rebuild his or her herd. And it turns out how this virus infects pigs is well known and has been known for decades. And it's one protein that sits on the outside of certain cells that the virus attaches to, and that's how it sneaks in and infects the pigs. And so what we're doing with gene editing is just deleting that one gene that makes that one protein from the pig genome so that the virus has no way to sneak into these cells. And the work that we and our collaborators primarily have done so far suggests that this is probably going to work. So it's certainly a very new area in terms of science, but we're very excited about it.
Thank you. So Rachel, on my more cynical days, I think that the press is enamored of CRISPR because of the name. But then when I think about it, I realize that in fact it allows scientists to alter genomes in a way that previous technologies did, but much more slowly, much more kludgely, et cetera. What is it about CRISPR that has made it take off in a way that, for the scientists in the audience, zinc fingers and talons just kind of didn't?
Yeah, that's a great point, Sharon. CRISPR is not the first gene editing technology. Gene editing has been around for probably a couple of decades, depending on how you count. But up until CRISPR, you really needed to have a PhD in genome editing to do genome editing. The ways that the scientific community could manipulate the DNA were just a lot more complicated. You had to design a bespoke protein for each new site that you wanted to go after. And there were, and still are, only a very small number of people in the world who have that expertise. And so it meant you either had to be one of those people or pay a lot of money to one of those people to be able to do genome editing. So the technology was around, but was not broadly accessible. And I think it created this pent-up energy and lots of ideas for what you could do if you had access to that. And CRISPR, I think, changed everything. This is an overused phrase, but I'll use it anyways. It really democratized genome editing. Because as you saw in the video, it's quite simple to reprogram the system. The big lump of protein called Cas9 is the same in every experiment. So you can just buy a lot of that, put it in the freezer. You're not constantly making a new protein. And to target a new site in the genome, meaning to edit a new site in the genome, you just have to design a new piece of RNA, which if you're an academic researcher in a lab, you can order from a company custom. It'll show up in a couple of days, and depending on how you ordered it, it costs you tens to hundreds of dollars. And so all of a sudden, this became accessible to a much broader research community.
So we're going to circle back to the question of whether democratizing genome editing is an unadulterated good. But before we do that, let me ask Rachel one more thing about the pig work. So by CRISPRing out the porcine viruses, the PRRS, as you pointed out, that could save herds and could be a huge benefit to livestock producers. A couple of decades ago, when there was some hope for even discussion about raising pigs as organ donors for humans, the FDA shut that down because of the concern about the porcine viruses that could in the liver, in the lungs, in the heart, whatever, if transferred to a human recipient, could cause very deleterious consequences. So you described the pig work in a livestock setting, but I'm wondering if the technology, the approach that you're describing, could also turn pigs into a source of donors for people who are otherwise on the waiting list forever.
That's a very good question. I'll say it's not something that we or our partners are working on, but I'm certainly aware that there are others who are using CRISPR or other types of gene editing to try to modify pigs for xenotransplantation for exactly what you're describing. I think it's early days, but there are a number of companies who are investing heavily in trying to do this right now.
Let me move to our next speaker, who is Kevin Esveld, an assistant professor at the MIT Media Lab, where he leads a team of scientists called the Sculpting Evolution Group, which explores evolutionary and ecological engineering. As a grad student at Harvard, Kevin helped develop CRISPR-based genome editing, and in 2013 recognized the potential for CRISPR to allow a process of directed evolution called gene drive. His scientific philosophy includes a pledge that he puts this way: as scientists, we have a professional responsibility to share the possible consequences of our research with the public in an understandable manner. More generally, we must invite, listen, and respond to concerns as best we can. We are morally responsible for all consequences of our work. And Kevin has some slides, so take it away.
I'll talk for a little bit first. So I'm middle-aged in CRISPR terms, in that I started working with CRISPR when Cas9 debuted in 2011. But I came about it through a roundabout way. I was never that into genome editing per se. Rather, I had amazing parents who took me to the Galapagos Islands when I was in the 6th grade. And I was just astounded by the incredible diversity of life. I wanted to understand how evolution made such incredible things. And that got me interested in genetics, but through the lens of evolution. I was always interested in how did different organisms interact in ways that led them to evolve new interesting traits. Could we learn how to do that? So for my PhD, I worked with David Liu at Harvard University and built a synthetic microbial ecosystem in which we essentially harness a virus that infects bacteria, which evolves very, very, very quickly, to evolve things that we want. And we did this because I'm fundamentally lazy, and I would much prefer that to be able to set up a system, hit start, and then come back a while later and have it spit out the answer. And that's what this does. But when I moved to my fellowship at the Wyss Institute, where I worked with George Church, there were people who do DNA origami, where they make lots of DNA and then they fold it up into complicated shapes, including molecular machines. But they used the same virus that my system used. And they would make it in these 10-liter batches, and so there you can just envision the virus floating in the air. And so no matter how hard I tried, the virus would always infect my cells before I was ready. And then I heard about this amazing CRISPR system, and I started playing with CRISPR in 2010 with the native system for my bacteria, which doesn't work very well. And then this amazing paper came out about Cas9, and I thought I'd try it, and it worked on the very first try. Let me immunize my cells against the virus that I didn't want. And then a subsequent paper showed that Cas9 could be retargeted and would work in a test tube and therefore possibly in mammalian cells. And one of my good friends had been working on some of the older genome editing technologies, said, hey, wait a minute, you've been working on this. And I said, yes. And he said, well, we should collaborate and do it mammalian genome editing. And I said, why? Jennifer and others have been working on it for some time. And he said, because if we stumble across even the tiniest little piece that someone else would miss for another few months, this is going to be so big that it would be worth it to the world. And he was totally right. So we had a fun for about 6 months, and then I decided, there's enough people working on CRISPR, it doesn't really need me anymore. But then I was walking through the Emerald Necklace in Boston, and I wondered, are any of these organisms out here ever going to be edited? And the answer is probably no, because when we edit an organism, we're editing it for our benefit. We're diverting its resources away from survival and reproduction in the wild, which is why no selectively bred or engineered organism has ever outcompeted its natural counterpart. But then I thought, well, could we use CRISPR to overcome that? What if instead of editing the genome with CRISPR, we encode the editing machinery itself in the genome? So here, imagine that you have a mouse and its genome, and there what you deliver in is a piece of DNA that encodes the change you want to make and the CRISPR system responsible for making that change. So CRISPR cuts the DNA, and the cell incorporates the new sequence. Now this cell knows how to do CRISPR genome editing on its own. It cuts the other chromosome and copies itself over. So now this reproductive cell in this mouse has 2 copies, which means when it mates, all of the offspring are guaranteed to inherit one. And in the reproductive cells of those offspring, CRISPR edits the original copy. So now the offspring have 2 copies. When they mate, their offspring are guaranteed to inherit. And editing happens again, and again, and again. So by programming CRISPR to edit the original version and replace it with the new one in every generation, we can cause that edit to spread through an entire wild population. Now this is a phenomenon called gene drive. Evolution discovered it long ago. Evolution just gave us all our best tricks on a silver platter. And CRISPR lets us harness this ubiquitous natural phenomenon called gene drive. So gene drive is the phenomenon of a gene spreading through a population very reliably, even if it doesn't help the organism reproduce. And this is critical because almost nothing that we would want to do to a population, immunize it against disease so it doesn't infect us, for example, that's a cost to the organism. But with a gene drive system, we could alter that wild population anyway. On the flip side, you've heard about how easy it is to use CRISPR. It is an experiment that in principle one person can do. If you make if you make a CRISPR-based gene drive, and you let that organism go, it takes appallingly few of them released into a wild population for it to start spreading. And this version will keep on spreading. We really don't have the institutions to govern this kind of technology, or at least not in the context of technology. If we develop a new drug, goes through FDA approval, your doctor recommends it to you, you can say no. If we develop a new gene drive system to edit the environment in your community, even if it's put up for a vote, if you vote against it and you're outvoted, it's going to affect you anyway. This is more like passing a law or infrastructure. It's a community-based shared decision because we all rely on the shared environment. So most of my career since then has been trying to come up with ways of ensuring that this research is open from the very earliest stages, which is completely counter to the way science is done. Now you might imagine if you were setting up science from scratch, you probably wouldn't encourage all of these taxpayer-funded academics to keep everything they're doing a secret from one another. That's pretty clearly not the most efficient way to make discoveries, because you get a lot of needless duplication, you don't know who to collaborate with. It's also, by the way, miserable because you're always afraid that you're wasting your time because 5 other labs are doing the same thing, or in the case of CRISPR, several dozen other labs. But that's the way it is, because science evolved back in a time when the fastest way to share your results with your colleagues was to write them down on paper and send them off by horseback and sail. And we really haven't updated the system much, even with the internet, even though the cost of information sharing is free. So my hope is that this one field can be a trial for changing the way we do science. Because here, with gene drive, if you even begin to develop a system to alter the shared environment, and you do it the traditional way without telling anyone, you're denying people a voice in a decision intended to affect them. And I think that's wrong. The National Academies apparently agrees, but that doesn't mean that the incentives in science are changing. So that's what we are working to change, and our hope is that if we can do that in this one area of science and show that it works, then maybe it will spread to others.
And Kevin, tell us, if you would, a little bit about one example of the kind of transparency, openness, democratization that you're talking about. You have made several trips to, I think, both Martha's Vineyard and Nantucket. What is the project that you have told residents there about, and how has that gone?
Well, first, can you please raise your hand if you've been bitten by a tick? Yeah. How many of you know someone who's had Lyme disease or babesiosis or something like that? Yeah, okay, we're in the Northeast. It's actually the most common vector-borne disease in the United States, is Lyme disease. People are afraid of the mosquitoes, but it's the ticks that are going to get you. So I was interested in this question of how should we be developing these kinds of technologies? And so I thought, well, Lyme disease is the local problem that we could address that might be serious enough to justify editing a wild species. And in this case, none of you probably particularly want to be bitten by a genetically engineered tick, particularly because even with gene drive, we'd have to release a lot of ticks into the environment to get anything done soon, so that's probably a non-starter. But the ticks aren't born infected with Lyme disease. They get infected when they bite an infected white-footed mouse, usually. There are other reservoirs, but that's the most important one. So the tick bites the mouse, gets Lyme disease, and then it bites something else and transmits it to that. That could be another mouse, or it could be a child. I'm from the West Coast. This notion that there's lots of ticks out there is just horrifying. There should not be ticks in the woods. Who came up? Evolution, what are you doing? And the notion that they give you disease, of course, is even worse. So the fact that our children often are not allowed to run freely through the woods, at least not without nightly tick checks, is just awful. So the question was, could we heritably immunize the white-footed mice in order to block transmission at the source? But this is not the sort of thing that you want to edit all of the white-footed mice. So in fact, we went to the communities on islands, not to propose a gene drive, because that kind of gene drive would spread, the mice will take the ferry too. It would spread back to the mainland and affect all of the white-footed mice. So we wanted to start with something smaller. So we went to Nantucket in the vineyard to just say, how about we just engineer mice to be heritably immune to Lyme disease? And these are islands. If you introduce enough copies of those genes, then you're not going to be diluted by billions of other mice from all sides. That will protect the populations on the islands. Ideally, no infected mice, no infected ticks, no infected kids. And we went there first to Nantucket before we had any funding, before we'd done any experiments in the lab, when this was just we think this is possible. This is an engineering challenge, not a scientific challenge. And that, in turn, was only possible because of CRISPR. Only when you can reliably edit the genomes of lots of different organisms can you be confident that you could edit the genome of, say, a white-footed mouse, which is not exactly a model organism. And so we went to the communities and said, are you interested enough in this for us to bother? If so, how should we go about it? Should we immunize them against Lyme, against ticks, against both, or neither? And how do you want it introduced? Do you want us to do this in a way that is as natural as possible? That is, we can take the acquired immunity that mice naturally get when they're exposed to these pathogens and just encode that in the genome using CRISPR, but it would come originally from white-footed mice. Or we could skip a lot of steps and use the pre-existing immunity genes isolated from laboratory mice and house mice, but that's a different species. And the community broadly said, yes, please. This is bad enough that we want you to at least start the research. And if you're going to do anything, you may as well immunize them against everything. But we would really rather that you not move genes across species if you can help it. So try to keep it as natural as possible. Now, natural is a very complicated word. We can talk a lot about what natural means and does not mean. We made it very clear that these would be genetically engineered mice. Mice do not naturally pass immunity on to their offspring in this way. But it's something that we think that we could do and the community is interested in, and so we now regularly go back to check, and the project, which is now called Mice Against Ticks, is now a couple of years in. And honestly, we've probably gotten a lot more media attention than we deserve, given the amount of actual laboratory progress we're making, but our hope is that this can be a model for how this sort of research should be done, where you go find an interested local community that might be early adopters and ask them if they're interested and how to go about it. So there's steering committees on Nantucket and the Vineyard that are guiding the project. It's not about governance, because governance, there's plenty of local governance mechanisms. New England town hall democracy. That's how decisions get made. But the steering committees are there to tell us what the community members think about the project, what they understand, and what they want, and to update us in case people decide that they're changing their mind based on what they hear. We want these technologies to be developed in a way that people are comfortable with, and we want to hear everything that anyone imagines might go wrong. Because we know that people understand their local environment better than we do, and when you're altering an ecosystem, really when you're engineering anything that evolution made, you want to start small. You want to make the smallest possible change that you think can solve the problem. And you want to test that on a local scale before you think about scaling up. So that's why we're starting without any form of gene drive. And then ideally, we might be able, if it works on Nantucket and the Vineyard, we might be able to introduce it to the mainland using a localized form of gene drive that we're developing that would alter the mice only within a town's boundaries. And then natural selection would favor the immunized mice within the boundaries and eliminate it on the outside. The trick is you just have to teach the genes to vote.
Even though this is not your work, I can't resist asking you to comment on whether you think the use of gene drive against malaria has a realistic hope of happening.
Yes, so the idea of harnessing gene drive has been around for a long time before CRISPR. It was just CRISPR that made it possible and made it accessible. And there's a wonderful group of scientists based in London called Target Malaria that have been developing a drive system against Anopheles mosquitoes, which are responsible for about 500,000 deaths a year, most of them children under the age of 5. In the time that we've been talking to now, about 25 children have died of malaria. What if we could alter those mosquitoes so they can't spread disease? Or what if we could crash the population to a low level, say 5% of current levels? Then that wouldn't be enough mosquitoes to efficiently transmit it person to person. And then once malaria is eradicated, then we could let the mosquitoes come back. But yes, if you're even more concerned about the prospect of a technology that lets people single-handedly edit an entire species, it also lets us crash a population. Because imagine that the edit we make turns them all into males. So as to whether or not that works, I have to be incredibly careful about what I say, because malaria is such a horrific problem, and it can only be solved by the full power version of the drive system, because the logistical challenges in Africa are so great and there's so many mosquitoes there. But that requires every affected country to agree to genetically engineer all of the mosquitoes. And that is a diplomatic challenge, to say the least. So I am hopeful, but it might be a long time. Technology is no longer the limiting step.
Okay, so as you've heard from Rachel and Kevin, none of this work raises any concerns at the policy level or ethical level, but we've asked Robbie to join us anyway. Robbie Barbero is the Chief Business Officer at Ceres Nanosciences, which is a diagnostic biotech that, as coincidence would have it, is developing a non-invasive test for Lyme disease. Is that still part of your thing? Okay. From September 2012 to January 2017, Robbie worked in President Obama's White House Office of Science and Technology Policy, where he developed and implemented policy on life science issues, including the BRAIN Initiative, the Precision Medicine Initiative, and genome editing. So Robbie, tell us, if you would, about how the work that we're hearing about this evening intersects with policy considerations.
Great. Well, thank you very much. So I am actually a total CRISPR neophyte. All right. So I do have a PhD in bioengineering. However, I got my PhD 6 months before the first CRISPR paper was published. All right. So I thought I was, and I had spent 12 years at the bench doing research before that. I thought I was extremely knowledgeable and relevant in my own field until 6 months after I left the field. The whole thing got turned upside down. So luckily, my co-panelists here have already talked about a lot of the technical capabilities and the interesting science here. But what I'm going to do is try to describe to you what it was like to be a biological engineer with over a decade of laboratory experience in commercial and academic research settings, who had the privilege of working in what was arguably the most pro-science administration ever in American history, at a time when a once-in-a-lifetime discovery is made in your very own field, okay? So I didn't pick this, I happened to be the one who was sitting there when it happened to me. And the way I'm going to try to illustrate this for you is to walk through a timeline of events. So what I'm going to try to do is weave together, and this is all publicly available information, right? So if you're hoping for another tell-all, I'm not a novelist, I don't have anything there. This is just publicly available information. But it's the advances in bioengineering that we've been discussing today, activities that the administration was working on related to biotechnology and biosciences, and because the purview of issues in the White House are everything in the entire world, some of the world events that were occurring at the same time, which were beyond our control, unrelated to bioengineering in many cases, but in many cases, totally sucked the oxygen out of the room for some of the stuff that we wanted to be working on. Okay, so I've never done this kind of timeline approach. If you all start falling asleep, I'll skip a few steps, but let's get started. Okay, April 2012, put down my last pipette. All right, done. Have not touched a pipette since then. No more lab work. Summer of 2012, moved to DC for the Science Policy Fellowship that I got to go work in the White House. When I joined the White House, I asked my boss what I was going to work on, and he asked me if I was familiar with Richard Hamming. Does anybody know who Richard Hamming is? Okay, so he was a researcher at Bell Labs, the amazing place that generated all sorts of innovations in the middle of the 20th century. He used to ask his colleagues, what are the most important problems in your field? And they would answer. And then he would say, what are you working on? Apparently this would annoy his colleagues because it would force them to confront the fact that they were working on something that actually wasn't that important. So imagine me, right? Just a few months before, I had been worried about what concentration of salt to use in the buffer for the experiment I was going to run the next day. And now my boss was telling me, someone with no prior policy experience, that I had to find the most important policy problems in my field, understand them well enough to figure out what some solutions could be, and then be ready to brief the President of the United States on them. Okay, so August 2012, first CRISPR paper published. I didn't read it. December 2012, genomic cruise missiles makes the lists of Science magazine's top breakthroughs of 2012. But to be honest with you, that whole section, it didn't make the top, it didn't win, right? It was one of the runner-ups. And that whole section is focused on zinc fingers, which were discovered in 2002, and talons, which were discovered in 2009. The article talks about how researchers have made engineered pigs that are useful for studying heart disease. And then it briefly covers CRISPR, and it says it might hold promise as a replacement for talons at some point in the future. I did read that. February 2013, a paper demonstrating the use of CRISPR in human cells is published. Okay, now it's starting to get real. I didn't read that paper either. April 2013, President Obama announces the launch of the Brain Initiative, which is focused on developing new technologies to help understand how circuits of neurons lead to the emergent properties of the brain, like memory and learning and cognition and health and disease. Since then, it has grown into a $1.5 billion initiative that has received bipartisan support. Starting in early 2013 and continuing for the next 4 years, I actually did what my boss told me to do. I started to talk to as many people in my field, in government, in industry, and academia, saying, what are the biggest problems in our field? What is not happening that should be happening? What are the things that need to be done? What could the government do? Lots of ideas. Got a lot of input, a lot of great ideas. Around the end of 2013, right? So it takes a long time to get the most important people in your field on the phone and ask them good questions and try to understand what they're saying. Near the end of 2013, the unreadiness of our biotech regulatory system for the future products of biotechnology starts to rise to the top of this list of feedback that I get. Although I will note, nobody in that entire year told me that CRISPR was going to change everything. Okay? So this is a full year after the publication of the first paper, and at least half a year after the publication of the second paper, and nobody is telling me this is going to change everything. There's lots of discussion about cheap DNA sequencing, DNA synthesis, some discussion about talons, all right? So I'm still trying to figure out what are we supposed to do? Is there anything to do here? So spring 2014, that is the point when the number of publications pertaining to the use of CRISPR actually surpassed the number of publications pertaining to the use of talons, right? So one of the things you're trying to figure out when you're sitting in a large organization is, what is the problem? You know, is this a real problem, or are people is it a shiny object that people are chasing for no reason? And so looking at publications is a great way of doing that. So it wasn't until nearly 2 years after that first publication that the scientific community started to show through their own output that this was a real thing. Okay, so right around that time, I initiated an effort internal to the US government to begin evaluating the readiness of the regulatory system for future biotechnology products. Then, in the June of 2014, Edward Snowden gives thousands of stolen documents to WikiLeaks, which end up totally dominating the news and all of the attention of everybody in the United States government for months. August 2014, CRISPR for gene drives first proposed in Science magazine. August 2014, James Foley is beheaded, and the video is released. ISIL and the caliphate continues to grow, in part by disseminating propaganda through US-based social media and tech tools, right? So that, you would think it wouldn't matter to OSTP, but guess what? They're using our technology to move things forward. So all of a sudden the White House is saying, what are we doing? What are we doing? Not my portfolio, but still taking up a lot of oxygen. August 2014, the WHO declares a public health emergency for the largest Ebola outbreak in history. It starts in West Africa, but it reaches Europe and the USA. Tens of thousands of people and ultimately end up being infection. It makes it to the US. It lasts all the way till 2016. And remember, I'm supposed to be running an internal effort to figure out what we're going to do about biotechnology and the products that are coming. October 2014, government shutdown. Healthcare.gov crashes. January 2015, we launched the Precision Medicine Initiative, which grows into a $1.5 billion initiative with bipartisan support. February 2015, scientists call for an international discussion about the use of gene editing in humans. So this is almost 3 years after the first paper, and now the scientific community is saying, hey, guess what? The government should do something about this. Spring 2015, civil war in Syria totally dominates the news. Spring 2015, Russia invades Ukraine. That takes up a lot of attention. March and April 2015, the National Academy of Sciences, the Chinese Academy of Sciences, and the UK Royal Society plan an international human gene editing summit. Around the same time, the White House and the NIH both articulate that human germline editing is a line that should not be crossed for now. July 2015, finally, we're able to publicly announce that we are going to be initiating a set of activities to modernize the US biotechnology system. And it's actually not even focused on humans, it's focused on plants, animals, and organisms. And we have a whole plan, three different activities we're gonna do, multi-year effort. This is just the start of the process, right? So it's taken us this long to just get to be able to say we're gonna publicly do this. And then in the fall of 2015, Zika outbreak. Rapid increase in microcephaly cases. By December, WHO connects Zika and microcephaly, and the first microcephaly case in the US happens in January of 2016. December of 2015, Human Gene Editing Summit happens. Remember the one with the Chinese and the UK and the US? And there's a statement of principles released by the scientific community about what the appropriate use of gene editing in the human germline is, right? So whether we can pass it on. January 2016, the administration announces a cancer moonshot initiative. Remember, Vice President Biden's son died of cancer, and so there's a push to do something around there. It ultimately grows into a nearly $2 billion initiative with bipartisan support. February of 2016, gene editing is put on a list of weapons of mass destruction by the Director of National Intelligence in the annual Worldwide Threat Assessment released by the United States government. I didn't see that one coming. June 2016, the White House Organ Summit is something that we organize out of our team, where money and policies are announced focused on reducing the organ transplant waiting list, including, as you mentioned, the use of biotechnology as a potential future way to address that. So now this is, we're almost at the end, remember? 2016, the clock is counting. We're down to days, not years left in the administration. And we still haven't done any of the things that we said we were gonna do in this space. Finally, in September of 2016, we released a 5-year strategic plan, and we put out a draft of the update to the policy that the federal government uses to regulate products of biotechnology. However, because of the fact that we live in a democracy, and we feel like input from experts and from the public is important before you finalize any rules and laws, we allow the public to comment on that. So we've just put this out in September, and now we have 4 months until we walk out the door to get those comments, incorporate them into the final version, and finalize it. In October of 2016, we have our final big science event with President Obama. We host a Frontiers Conference to explore the future of innovation across 5 themes. I have the privilege of being one of the people who gets to organize the medicine and healthcare track. Kevin gets invited to come talk. The plenary features President Obama and several guests talking about the future of medicine and healthcare. And we actually timed this with the release of an issue of Wired magazine that's edited by President Obama. Then in January of 2017, we finalize our update to the federal policy around how biotech products are going to be regulated. And I leave the building 6 days later. All right, that's it.
Are there questions from the audience? In the front.
I would like to ask each of you what you think the future is, and the future I'm defining as the next 3 years. Do you want to go first?
Sure. I'll narrow my answer to where gene editing touches the world in the next 3 years. And I think in the next 3 years, we're going to see it, tip of the iceberg, begin to transform medicine. Already today, there are open clinical trials where patients, a small number of patients, are beginning to be dosed with therapies that are using CRISPR gene editing to treat sickle cell disease, for example, or to treat certain kinds of cancer. There have been for many years other trials evaluating other kinds of gene editing technologies. And so I think this is the beginning of what will become an entire new wave of medicine. You know, as you think about medicine today, really you get prescribed a small molecule, usually in the form of a pill, or an antibody. Those are really the 2 pillars of drugs today. And I believe that within a fairly short period of time, there will be 3 pillars, and the 3rd pillar will be gene and cell therapies. And gene editing will be an important component of that.
Robbie, I loved your overall description because it illustrates what is wrong with the way we approach the world is that we get distracted by these momentary political crises when the way we live today is overwhelmingly shaped by technology. And it is future advances in technology. It is the technologies that we invent and our wisdom in deciding whether, when, and how to use them that will overwhelmingly determine the future of our civilization. And that means we need to talk about how we go about doing that. The next 3 years, I'm hopeful that we might see some change with that, although it's probably not necessarily for good reasons, because we have increasingly access to these technologies is very widespread. We've already had certain rogue scientists in the news for doing unethical things. And as you start getting technologies that can unilaterally affect large groups of people, combined with our tendency to get distracted by momentary concerns, I think we're probably going to be forced to think a little bit harder about the way that we develop and use technology.
I will just say that I think we will see an increasing number of plants and animals and microbes that are being genetically engineered. Whether those make it into widespread, release into the marketplace or consumer applications remains to be seen. But my company doesn't do genetic engineering or any of this stuff, so this is not based on inside knowledge. But my read on all of this is there's so many people and companies working in this space, so many products being developed, so many applications being pursued. I think you will see an increasing number of things showing up in every part of your life that have been gene edited by technologies like CRISPR. And it'll surprise you, things that you didn't even expect would come from gene editing.
And if anyone needs a little light bedtime reading, I urge you to look up CRISPR patents and see how many are being filed. A question here on the aisle.
You referred to the rogue researcher in China who did the editing of the babies for HIV. I just would be interested to hear, because of the democratization that you mentioned of the use of this, and you go to places where, quite frankly, there was a red line about germline editing, and yet somebody crossed that line. How do you actually protect ourselves against sort of rogue scientists? And as you go to things like GMOs and changing the food source, I mean, how do we sort of recognize your choices that you mentioned, Kevin? You know, if I want to eat a chicken who's been modified or a piece of pork that's been modified versus not wanting to eat it, how do you actually protect the identity of the food chain?
Maybe that's a Kevin question because it has to do with public input, et cetera.
Yeah, so there's not too much we can do about individual rogue scientists beyond changing policies, but that is challenging because it's at the international level.
And there's no mechanism for changing international policy.
No. So
let's just be clear about that, right? And the current political environment is taking us away from the ability to influence international policy.
Although that is actually an excellent opportunity to engage in what we call scientific diplomacy, in that we can still talk to our colleagues in other nations even if the governments won't talk to each other.
But you can't make someone do what they don't want to do.
No. And right now the incentives are not pointing in good direction, so if we want to see something different, we need to change the incentives. In the case of, say, gene drive, it's fortunately a technology that is not readily weaponized. That is, it's slow, it only spreads over generations. It's obvious if you look for it. If you sequence the genome, you can't miss it. And once you see it, the very first thing we made when we tested it is we made one that would make a change in yeast, and then we made another one that would undo that change, that would overwrite the first one and restore the yeast back to the way it was before. And that works. So anything that's slow, obvious, and easily countered is not much of a threat. But most technologies are not like that because entropy. There are more states of civilization not being in a good way than there are civilization working in ways that we're happy with. And so increasing applications of force allow people to spread it in unwanted directions.
I would also add here, I mean, I think, so from a policymaker's perspective, you can't fix every problem, right? You have to prioritize. I mean, it's just, there aren't enough resources there. So I was always trying to prioritize. Where are the things where there's the greatest risk, the largest possible harm to the most number of people? And so your question was a little bit loaded because you bundled things across that spectrum all together into one question. And that is a policymaker's nightmare, right? Because addressing the totally legitimate and appropriate desire of a consumer to choose what they eat, all of which is safe because it's all been through our regulatory system, is different from whether someone is genetically engineering a human in a way that change will pass on throughout humanity or
Whoa, hang
animals.
on. Let's put this in perspective.
Yeah.
If people edit their children now, if everything goes well, those children will have vastly better technologies for editing their own children. So no, I don't buy this. You edit the germline now and it's a forever thing, unless civilization crashes in the meantime.
I agree
and since
with
that's
that.
the lost scenario, we're not going to focus on that.
Yeah, and then animals and microbes. I mean, all of these things there are different time scales and different area scales and impact scales, and so you have to. Anytime. So let's imagine you came to me when I was working in the White House, right? So you came in with the meeting and you said, here is the thing that I'm worried about. And if you asked me a broad question like that, I would have asked you about a dozen follow-up questions to narrow down to what is the thing that you are actually truly most worried about. And then as soon as we figured out what that was, I would have said, great, what do you think the answer is?
Question on the far right. Yes, sir.
You know, when we first broke the atom apart, we didn't try to shove it into the Department of Electricity or whatever was there at the time. We created a whole new agency to cover this. I'm worried about rogue actors, but I'm sort of worried even about well-intentioned actors playing Jenga and pulling the wrong thing out of the thing. I almost feel like it's not an FDA problem. It's not an agricultural problem. It's like a fundamentally new agency type of problem in my mind. And so if I advocate for that, or I say halt until further proper
So what is the problem?
The problem for me is the unintended consequences. You know, if there's a overrun of white mice in Nantucket soon, what's going to happen? You know, I have to be honest with you, the benefit of slightly cheaper bacon right now is not making me feel better about pulling the wrong thing out of the wrong thing.
So
Just to be honest.
So
I almost feel like we need a much broader, bigger oversight, new agency possibly that crosses functions. That's how I feel right now.
So can I give an anecdote maybe? So I don't come from a plant background at all. I knew nothing about plant biology until I took this job and started learning from some of the biggest agriculture companies on the planet, some of the smallest agriculture companies on the planet, where our food comes from. You want to talk about transparency? I think your average consumer has zero idea where their food comes from. There's some great pieces online and in other resources that you can find that show the plants we eat today and what they used to look like, and talk about how we got from where we were to where we are today. And the most traditional breeding technique that's used today is to take seed or other immature embryos, basically, of a plant, throw a bunch of chemical mutagen on it, grow up a bunch of those plants, see what happens, pick the ones you like, and breed them for years to try to get kind of exactly the right function, shape, quality that you're looking for. So what we've been doing to our food for years is massively rearranging the genome every generation without any idea of what we're doing to it. And that seems to have gone shockingly well. And I personally like the idea of now being able to come in and try to be a little more intelligent about it and you know, use a scalpel instead of the A-bomb to try to manipulate the genome. And so I think there's a huge challenge of how we put this conversation in context, and I think very few of us have access to that context. Your question earlier, I think, is perfect. I love the idea of consumers having access to information about what they're eating. We don't have that today. You know, I went to Whole Foods last night to buy some bottled water for my hotel room, and one of the signs, one of the labels next to the bottled water said, non-GMO. There are a lot of problems with that I think this room can appreciate. There are obviously no organisms to be or not to be genetically modified in the water. And if there are, that's a totally separate situation. But that's the information that consumers have today. And so I think it's really hard to put this conversation in that context.
Sir, toward the back.
Hi, following up on the conversation, what do all of you think about the USDA deciding to treat a gene-edited organism or food differently from a GMO food?
From a technical perspective, if you give me only the ability to knock out existing genes, the suite of changes that I can make in that organism is dramatically more limited than if I can move in any useful trick that some other organism has created. So in the sense of what is possible, it's a perfectly reasonable boundary to draw, in particular because when you throw a ton of chemical mutagens at it, you're going to knock out all kinds of genes, and so if you can get a phenotype that way, you can get a phenotype with CRISPR.
By phenotype, what do you mean by phenotype?
A particular trait that you're interested in. Because what we're talking about in gene editing, the USDA has decided that if you use CRISPR to go in and just knock out a gene, like the first application in the video, then that does not need to be treated as a genetically modified organism, because loss of function mutations occur in all genes all the time, especially if you bathe them in chemical mutagens the way we've been doing for the last 50 years. And there's also, but controversially, the FDA has come up with the opposite decision for animals.
Wait. This is getting, now we're mixing pots here. So I think it's important. So let's stick with plants. Plants that are going to be eaten are regulated by at 2, potentially 3 federal agencies. USDA makes sure they're safe for the agriculture system. Okay, so the decision you're talking about is an agriculture decision only. Now, if you're going to feed that crop to animals or humans, you have to go to the FDA, because that's the food part of it. And the FDA will ask you to prove that it is safe for humans or animals to eat. Okay, so that doesn't mean that these crops are now completely without oversight by the United States government. If there is any pesticidal function associated with the genetically engineered crop, you then have to go to the pesticide agency, okay? So I do want to clarify that. Now, the part you're talking about is the use of CRISPR in genetically engineered animals.
Yes, and it's noteworthy that different agencies made different decisions on that call. But my point is
because
that
they have different responsibilities assigned to them by the United States Congress.
what we're talking about is duplicating a pre-existing mutation in such that on the molecular level, it is identical to what you create through chemical mutagenesis.
But you would agree that plants and animals are different?
I certainly don't agree that you should treat gene editing in the two separate categories of organisms as differently based on any kind of spectrum of risk.
Ooh,
no.
This is good. Now we finally found something we disagree on, Kevin. I'm so happy. We definitely do not have time to flesh this one all the way out to the end.
Also, on the priority spectrum, this is very, very low. This is not the sort of technology we need to worry about for the future of civilization. I understand people are passionate about it, but no.
Question on the far left.
Hi, my name is Jade Lovell. I'm part of NYSCI's Alan J. Friedman Center. And I run a program that helps train young people to communicate science. I'm also a host on Discovery Channel, and we were one of the first people to wonder aloud in 2015 why this was the first time the scientific community had got together to have an international conference on human gene editing. So my question is to you, Kevin, and also to you, Sharon. How much of a role does the media play in educating the public on these new technologies, and do you think the media is doing a good enough job?
Kevin.
Oh,
no.
Definitely has to be you.
So the media can always do a better job. We are addressing a community whose scientific literacy is nothing to be proud of. So we are doing a great deal of educating that people like myself feel really should have been done when people were in K through 12 school. So there's a lot of catching up to do. Now, when you ask reporters, what do you feel your role is in terms of educating the public? The party line answer is 0. That's not our job. Our job is to inform. And we always try to pitch our stories at a level that we think our readers, who differ by publication, by whatever the medium is, a level at which we believe our readers will understand. We always try not to be irresponsible, and we're doing our best, but then things like CRISPR babies happen. Kevin, what do you think?
What do you mean by things like CRISPR babies happen?
So the second International Human Genome Editing Summit in Hong Kong last November looked like it was going to be a fairly kind of a snooze. There weren't going to be any new scientific results unveiled. There was going to be a lot of talking on very important issues, including community engagement, the different cultural attitudes toward genetic engineering and genome editing across the world. And then it was hijacked by Dr. He's announcement, and everybody just went crazy for that. So any other message out of the meeting got completely drowned out. So the best of intentions can be waylaid by events, but that's news. I mean, that's what we do. So I'm certainly not complaining, just describing. In the way back, a question.
Hi, I'm Natalie. I'm a kid. Question, could gene editing help with deformations of chromosomes? Okay, yeah, that's it. Okay, yeah, that's it.
Deformations of chromosomes. Rachel, can I
I'll
ask
give it a
you
whirl,
sort of
sure.
maybe a medical question?
Maybe someday is probably the best answer I can give you. Right now, the genome editing community is pretty good at deleting one gene at a time. As the video suggested, you can also insert a new gene. That's a lot harder, and we're only good at it at certain places in certain circumstances. To do anything much more sophisticated than that, like adding a whole new piece to a chromosome or maybe trying to remove an extra chromosome that's causing a disease, I think that's a vision that a lot of people have, but it's not a near-term technical reality. I can't tell you if that's 5 or 10 years or 50 years, but I do hope that that's where the technology will take us.
You guys were amazing. Absolutely incredible.